Hello everyone, welcome to Biotechnika's session on question paper discussion for the CSIR NET exam that happened in June 2024. I'm Dr. Nupur and I'll be taking you through Unit 6 questions that entirely deal with plant physiology. What we are going to do is, first we'll discuss the morning session questions and then we'll go to evening session, and each session questions will be divided in part B and part C. Right? So let's jump right into it. First, we'll be talking about the morning session questions, and in that specifically we'll be dealing with part B first. So if you've gone through the question paper, you would have noticed part B questions are pretty straightforward. I'll show the question first, we'll discuss the options, and then we'll also see the results, right? So every question will be followed by a discussion on that question. So, which one of the following represents the predominant source organ during phloem translocation in healthy plants? So in a healthy plant, which of these following acts as the main or the predominant source organ during translocation of phloem? That means during the transport of food. When the food needs to be transported, which of these acts as a source organ? Now you know that in plants, some organs are considered as source organs where photosynthesis is more, where food is being synthesized in large quantities. And then there are sink organs. Sink organs are where the food will be consumed. The food will be utilized because the sink organs are not photosynthetic, or photosynthetic rate is not very high. So they consume food. Sink organs consume the food produced by the source organs. Now if you look at the options, roots, developing fruits, immature leaves and mature leaves. So it's kind of obvious that out of these, the mature leaves are the ones which actively carry out photosynthesis. They fix more carbon than they give out carbon through the process of respiration, and that is the reason why mature leaves are the predominant source organ. Roots not involved in photosynthesis, so that's a sink. Developing fruits, non-photosynthetic parts, again sink. And immature leaves, even though some amount of some level of photosynthesis will be going on in immature leaves, but it's not sufficient for their carbon requirement. So when leaves are young and small, they act as sink, not as a source. Only when they grow and they develop and mature, that is when they can act like a source organ. So the answer here is option 4. Right? So the same thing is written over here, what I explained, that there are some organs, some parts of the plant that can act as source. These are the mature leaves, whereas the young leaves are going to act as sink till they become mature. Roots act as sink. Flowers, fruits, all such organs act as sink. Okay, so the answer for this question is option 4, mature leaves. Now the next question. Which one of the following correctly describes dark reversion of phytochromes? This is also a straightforward question. So if you've studied sensory photobiology in plants, you've studied phytochromes, this should be a very pretty question, easy question for you. So what is dark reversion? Phytochromes can exist in two forms, PR and PFR, right? The PR form absorbs red light and becomes PFR. PFR absorbs far red and becomes PR, right? So that is the conversion. They can convert from one form to the other. That's the simple cis-trans conversion. But in darkness, what happens? So let me explain that with a diagram. Okay, see, this is the PR form, the form of phytochrome which absorbs red light. So when it absorbs red light, which is at 660 nanometers, you can see that the PR is becoming PFR. It's becoming a form which can absorb far red light. PFR absorbs far red. You can see it's absorbing far red light over here at 730 nanometers, and it is reverting back to PR. Now, apart from this, there is another concept called dark reversion or slow reversion, which happens during darkness. Both these conversions, PR to PFR and PFR to PR, in presence of red and far red light respectively, needed some wavelength of light. Red light is 660, far red is 730. So these conversions happen in presence of light. But if PFR is kept in darkness, then it's a very slow reversion back to PR. If PFR gets far red light, then it's a very quick reversion. A very quick reversion to PR. But PFR under darkness, complete darkness, slowly reverts back to PR, right? So let's look at the question again. Which one of the following correctly describes dark reversion of phytochromes? Dark reversion is conversion of PFR to PR. It's a very slow conversion and it happens in darkness. Okay, so the answer is option 2. Alright. Yes. The next question. Again, a very straightforward question. If you know the concept, you should be able to answer this question. This is again dealing with, this is again from the topic of solute transport. Okay, solute transport in plants. So, which of the following is least suited for long distance phloem transport of photoassimilated carbon in plants? In very simple terms, the question is asking which one is not suited, which of the following is not suited to be transported in the phloem as food. The options are reducing sugars, mannitol, galactosyl sucrose oligosaccharides, non-reducing sugars. Okay. Now, again, if you know the concept, you will easily get to know the answer that in plants, the form of sugar that is transported is not glucose. Okay, glucose is not the form that is transported. Predominantly, the transport form is sucrose. Okay, predominantly the form that is transported is sucrose. Now, why is that? The reason for that is reducing sugars are not generally transported or translocated in the phloem. Reducing sugars like what? Reducing sugars like glucose, mannose, fructose. These are not transported in the phloem. Now, why is the reason for that? These reducing sugars have a free aldehyde or ketone group. This D-glucose has a free aldehyde group. Mannose also has free aldehyde group. Fructose has a free ketone group. Right? It's because of these free aldehyde or ketone groups that these sugars are reducing sugars and they are reactive. They are relatively more reactive. And that's the reason why it is not transported in the phloem. So reducing sugars are not generally translocated in the phloem. Then what is translocated then? Commonly sucrose. See, sucrose is a disaccharide of glucose and fructose. Glucose and fructose join together, but remember that it's a 1-to-2 linkage. That means both the groups, the aldehyde and the ketone group, are now actually bonded together in the glycosidic linkage. They are trapped together in the glycosidic linkage. So those groups are not free, and sucrose is a non-reducing sugar. Right? So sucrose is non-reducing. Then if you take sucrose molecule and you add 1 galactose to it like this, you take 1 sucrose and add 1 galactose, then this structure is called raffinose. Add 1 more galactose to it, then this structure is called stachylose. Add 1 more galactose to it, that structure is called verbascose. Right? So that is galactosyl sucrose oligosaccharides. Oligosaccharides which are made up of sucrose joined to galactose. So raffinose, stachylose, verbascose, they all come under this category, and it's okay for them to be transported. So plant does not have any problem translocating these galactosyl sucrose oligosaccharides. So this can be transported. Non-reducing sugars can be transported, and also mannitol. What is mannitol? Mannitol is a sugar alcohol made from mannose. Look at the structure of mannose. Mannose has a free aldehyde group. Mannitol, in mannitol, the aldehyde is now reduced to alcohol. Okay, the mannitol is now reduced to the aldehyde is reduced to an alcohol group. So because now the free aldehyde is not there, it's okay for mannitol to be translocated, and this form is also one of the options here. Mannitol is translocated through the phloem. Now the question is asking which of the following is least suited. Those would be the reducing sugars. As we discussed, reducing sugars have a free aldehyde or ketone group, because of which they are relatively more reactive, and that's why plants avoid transporting these reducing sugars. So the answer is option 1. Right. Yes, a little small announcement over here. So our enrollments for CSIR NET December 2024 and June 2025 exam is ongoing. You can see that there are many courses and allied courses also that is offered. We offer test series, we offer study material. So if you are serious about giving exam either for the December session of this year or June 2025, the number is given on this slide, and you can call anytime on this number and you can enroll yourself. It will be helpful because of all the courses and the material that is offered by Biotecnika. We try our best, we try our maximum to make sure that the process of learning goes smoothly for you. All right. Yes. So the next question again from part B. Okay, now this is from water transport. So the earlier question was from the solute transport part, and this now deals with water transport in plants. Okay, so the question is, a plant is not watered for 7 days continuously. From day 1 to day 7, the plant is not watered. Leaf and root water potential are measured every 2 hours starting from day 1 till day 7. Okay, so from the first day to the 7th day, they have measured both the water potential at the leaf and the water potential at the root for every 2 hours. Now again the question is asking which one of the following is least likely to happen. Okay, which of the following among the 4 options is the least likely to happen? So let's look at the options one by one. Pre-dawn leaf water potential declines over 7 days. Leaf water potential shows a diurnal cycle of highs and lows. Root water potential falls below leaf water potential at night. Root water potential fluctuates between day and night. Okay. Now to answer this question, you should know the concept of water potential. Now in very layman's term, water potential is the available water. Of course, it's not as simple as that, but you can just right now, if you don't know the concept of water potential, you can think of it this way. It is the available water, right? Now the plant is not watered for 7 days. So from day 1 you have stopped giving water. Overall the water potential of the plant is anyways going to decline. Since you are not giving it water continuously for 7 days, the overall water potential of the plant is going to decline, isn't it? Which means that over the period of 7 days, the leaf water potential will also decline, and the pre-dawn leaf water potential will also decline. Since the overall water potential is declining, the pre-dawn leaf water potential is also going to decline over the course of 7 days. Now, the other 2 points, that is point number 2 and point number 3, for that you need to understand that since the roots are more they are the ones who are in contact with soil, right? The leaves are in contact with air or the atmosphere. So roots are the ones which take up the water from the soil. And when roots take up water from the soil, it's usually the root cells which have a relatively higher water potential as compared to the leaves where the transpiration process is taking place. Transpiration process is taking place at the leaves, and from the leaves the water is going back to the atmosphere in the form of water vapor. So if you take any plant, whether it is properly hydrated or whether it is suffering from drought conditions, usually the root zone has relatively higher water potential as compared to the leaf zone. If you look at the values given here, root cells have a water potential value of minus 0.2 megapascals, which is relatively high because this is close to 0. Stem, you can see that it is decreasing. Leaf, it is decreasing, and atmosphere is of course the least one, right? And because of this gradient, because of this water potential gradient from the roots to the shoots, that the water can go in this direction from the roots of the plant to the shoot, right? So whether it's a well hydrated plant or whether it's a plant experiencing water shortage, in both these cases root water potential will be higher than leaf water potential. Now how much is the difference? That varies from case to case, it varies from condition to condition, but root water potential will be higher than the leaf water potential. And one second. And that is the reason why the answer is option 3. Okay, the answer is option 3. Root water potential falls below leaf water potential at night. That does not happen, especially at night, because at night time transpiration rates are relatively low. The roots will have higher water potential as compared to leaf water potential. So this one is the least likeliest to happen. Now what about statement 2 and 3? They can happen. Leaf water potential shows a diurnal cycle of highs and lows. Yes, because early in the morning the sun is not up, right? And at that time the transpiration process is less, As the day progresses, as the heat increases, as the sunlight becomes more stronger, the intensity increases, what happens is the transpiration process picks up. The rate of transpiration is more, and again it goes down as the evening comes. So yes, leaf water potential shows a diurnal cycle of highs and lows. Even root water potential fluctuates between day and night for the same reason. So 1 happens, 2 happens, 4 happens. It's the number 3 that is the least likeliest to happen, and that's why the answer is option 3. Yes, so coming to the next question. Which one of the following is a correct combination of 4 carbon intermediates formed during C4 photosynthesis in plants? So this question deals with the topic of photosynthesis, specifically C4 photosynthesis or C4 pathway. And again, this is a pretty straightforward question. So 4 carbon intermediates. Even if you don't know the pathway, but you know the number of carbons in each compound, you should be able to guess the answer here. So which are the 4 carbon intermediates over here? Malate is 4 carbon, oxaloacetate is 4 carbon, aspartate is 4 carbon. Alanine is not 4 carbon, it's 3 carbon. PEP is 3 carbon, and alanine and pyruvate are 3 carbons, right? So like if you go by this logic, the answer is option 1, malate and aspartate. But let me also explain the process. This may not be so easy every time. So you should understand the concept that during the C4 pathway, oxaloacetate is formed, but oxaloacetate is converted to malate. And malate is the one which is transported from the outer mesophyll cells to the inner bundle sheath cells. And there malate undergoes oxidative decarboxylation to produce pyruvate. Now, so oxaloacetate and malate, both are formed, both are 4 carbon intermediates, but it can also be converted to aspartate. Okay, oxaloacetate is reduced to malate by this enzyme NADP malate dehydrogenase in the outer mesophyll cells. But it can also be converted to aspartate by transamination with glutamate, and this reaction happens in the cytosol. So both oxaloacetate, malate, aspartate, these are the 4 carbon intermediates, but in the options, this is the one which is fitting. This is the one which is fitting. So malate and aspartate, 4 carbon intermediates. Answer is option 1. Next question. Which one of the following statements regarding plasmodesmata in a plant cell is incorrect? So question is asking about cell junctions, plant cell junction, plasmodesmata, and the question is asking which of them is not true. First sentence, they are specialized cell to cell junctions, that is correct. Plasmodesmata are junctions, they are small pore like structures which connect plant cells. So yes, they are specialized cell to cell junctions, that's correct. They are open channels that connect the cytosol of adjacent cells. That's also correct. If 2 cells are having this plasmodesmata connecting them, then there is cytoplasmic continuity. That means substances can move from cytoplasm of one cell to the cytoplasm of the next cell via plasmodesmata. Yes, so that second statement is also true. There are open channels that connect the cytosol of adjacent cells. Third, the plasma membranes of the adjacent cells extend continuously through each plasmodesmata. That's also true. The plasmodesmata channel is lined by the plasma membrane, which ensures continuity between the connected cell membranes. So this statement is also true, that the plasma membranes are connected. of the different cells via the plasmodesmata. It ensures continuity between the two cells. Fourth statement, plasmodesmata are extensions of chloroplast that interconnects the cytosol of the adjacent cells. Now the first part itself is wrong. Plasmodesmata are extensions of chloroplast. They are not extensions of chloroplast. Yes, they interconnect cytosol of adjacent cells. That second half part of the statement is true, but not the first part. So, which is the incorrect statement among all the four? It's option 4 or statement 4. Statement 4 is incorrect. It is a wrong statement. Now let's talk about part C questions from morning session. Yes. Now these are little more complicated, of course, and that's why they are 4-mark questions. And so let's go through the questions one by one. Following statements were made with respect to plant steroid hormones. First statement. The receptors for plant steroid hormones are found in the nucleus, similar to animal steroid hormones. There are multiple pathways for plant steroid hormone biosynthesis involving cytochrome P450 class of enzymes. The first plant steroid hormone was isolated from male gametophytes. Plants deficient for the steroid hormone brasinosteroid show underproliferation of phloem and overproliferation of xylem cells. Testosterone is a plant steroid hormone abundant in the vegetative tissues of the plant. Which one of the following options represents a combination of all correct statements? So they want to know which of these 5 statements is correct. True. Let's look at its statement by statement. The first one. Receptors of plant steroid hormones are found in the nucleus, similar to animal steroid hormones. This is not entirely true. This is not entirely true. For example, for brasinosteroids, the receptor is found on the plasma membrane. Okay, so this is the receptor. Brasinosteroid insensitive 1. BRI1. Which is the receptor for the brasinolides, and these are present on the plasma membrane, and hence that statement is that you cannot make that blanket statement. It is not completely true, hence statement A is incorrect. Statement A is not completely true. B. There are multiple pathways for plant steroid hormone biosynthesis involving cytochrome P450 class of enzymes. Now this statement is correct. If you studied steroid biosynthesis, brasinosteroid biosynthesis pathway, that you would know that it starts from campesterol, and then there are multiple pathways. There are multiple pathways leading to the production of brasinosteroids and brasinolides. Right? These are oxidation pathways. Both these pathways are oxidation pathways, and it does involve cytochrome P450 monooxygenase enzyme family. They catalyze most of the reactions of the brasinosteroid biosynthesis pathway via different routes. So the second statement is correct. Statement B is true, that there are multiple pathways for steroid hormone biosynthesis, and it involves cytochrome P450 monooxygenase class of enzymes. Third statement is the first plant steroid hormone was isolated from a male gametophyte, and that is true because first brasinosteroid was actually identified from rape pollen. Okay, the rape plant's pollen, from there, so pollen is a male gametophyte, and from there this hormone was isolated, the first steroid hormone. So C is also correct. Now, plants deficient for the steroid hormone brasinosteroid show underproliferation of phloem and overproliferation of xylem cells. Now this statement is not true. In fact, it's the exact opposite. What happens is the exact opposite of what is written. If you knock out plants for steroid biosynthesis, you create knockout plants for steroid biosynthesis, you'll find that there is over proliferation of phloem and under proliferation of xylem. So the statement that is given is completely opposite of what actually happens. So D is not correct. And the last sentence is castasterone is a plant steroid hormone abundant in the vegetative tissues of the plant. This is true. Specifically in tomato plants, it has been found that brasinosteroids are involved in the fruit development, whereas in the vegetative development, castasterone, which is also an active precursor of brasinosteroids, it is more abundant in the vegetative tissues of the plant. See, this is castasterone over here. This here is castasterone, which is an active brasinosteroid, and it has been implicated in the development of vegetative tissues in specific plants like tomatoes. So statement E is also correct. So the answer is option 2, which is B, C and E. These are the correct statements. Next question. Following are certain statements regarding CAM metabolism, again photosynthesis topic, and you have to find out which one of the following options represents combination of all the correct statements. See, most of the questions you will find it like this only in part C, especially from unit 6, where multiple statements are given and you have to find the combination of the correct statements or incorrect statements. So you have to read twice exactly what they are asking. So here again they are asking for correct statements. Okay, so the first one. The bicarbonate concentration is enriched in the cytosol during night by the CO2 coming in from the external atmosphere through the open stomata and mitochondrial respiration. Now, you should know that in the CAM pathway is predominantly seen in plants growing in desert or arid conditions, and for them, saving water is priority. Of course, they have to carry out photosynthesis, but one of the main concerns is conservation of their water, right? So, these plants, they keep the stomata open during night time. That is to make sure that water loss is not excessive, because if they keep the stomata open during the daytime, water loss will happen, right? So during night time, stomata is open, and during night time when the stomata is open, atmospheric carbon dioxide is taken in, it is converted into bicarbonate ion, and then it is fixed to oxaloacetate. So first sentence is actually true. Bicarbonate concentration is enriched in the cytosol during the night time from the CO2 coming from the external atmosphere through the open stomata and mitochondrial respiration. So this sentence is correct. Second, the oxaloacetate produced by the action of PEP carboxylase is stored in the vacuole during dark. Now this sentence is not correct. Oxaloacetate is formed. We saw that here, right? See, oxaloacetate is formed during the night time, but oxaloacetate is converted to malate, and it is malate that is stored in the vacuole, not oxaloacetate. So, statement 2 is wrong. Statement 3. During light, oxaloacetate produces malate that provides CO2 for Calvin-Benson cycle in the chloroplast. Now, this sentence is also not correct. Okay. What is wrong with this sentence? Oxaloacetate does not produce malate during the morning time. During light, when light is present, oxaloacetate does not produce malate. Malate comes out from the vacuole, and malate enters into the stroma of the chloroplast, where it is converted to pyruvate. Okay, so do you see what is wrong with that sentence? Oxaloacetate does not produce malate in light. Now statement D. During dark, Phosphoenol pyruvate is produced by the breakdown of starch present in the chloroplast. Yes, that is true. During darkness, you can see here, when the stomata is open, the starch which was produced in the and stored in the chloroplast, that is broken down to produce phosphoenol pyruvate. So that is the statement D. During dark, PEP is produced by breakdown of starch present in the chloroplast. So this is correct. And last, CAM is a mechanism of concentrating CO2 around rubisco by keeping stomata closed during the day. This sentence is also true. Both C4 and CAM pathways are CO2 concentrating mechanisms. Their purpose is to make sure that rubisco is surrounded more by carbon dioxide so that photorespiration can be minimized. So last sentence is correct. So A, D and E, these are the correct statements and the answer is option 4. Next, the nitrogen fixing bacterium Rhizobium leguminosarum, isolated from the root nodules of garden pea plant Pisum sativum, is cultured in a Petri plate containing appropriate nutrient agar medium. From this culture, a bacterial colony was picked up and inoculated into a liquid growth medium to scale up for the production of biofertilizers. Now, which one of the following statements is correct? Now, this is not combination of statements. We have given 4 options and one of them is correct. The rest all are wrong. Okay, so this is from the topic of nitrogen metabolism in plants. Okay, the first sentence. The liquid culture will be red or pink in color due to the accumulation of the pigment leg hemoglobin. This sentence is not correct. Why? Yes, the root nodule rhizobia, the tissues of the root nodules do appear red or pink in color. That is true. But those pigments of leg hemoglobin are not produced by the bacterium, they are produced by the host plant. So if the bacteria is growing on an artificial medium, can it produce the leg hemoglobin? No. That property or that ability is with the host plant and that's why statement 1 is not correct. Second, the rhizobial cells, when re-inoculated into the rhizosphere of a soybean plant, will effectively nodulate its roots to fix atmospheric nitrogen. So the sentence is saying that if you take these culture rhizobial cells, and you re-inoculate it into the roots of soybean plant, right? It will nodulate it. It will establish a colony there and it will start fixing atmospheric nitrogen on the soybean roots also. Now, what is wrong with this sentence? Nitrogen fixing bacteria show high specificity for their host organism. So usually one type of nitrogen fixing bacteria, one species of nitrogen fixing bacterium will be specific for a particular host plant. Now, since this rhizobium was taken from garden peas, Pisum sativum, it's very unlikely that they will easily colonize the roots of soybean because the host plant is now different, and like I said, they show host specificity. So, second statement is also not correct. The rhizobial cells cannot fix nitrogen when exposed to atmospheric air, and this sentence is true because the enzyme complex which is actually responsible for fixing atmospheric nitrogen, which is that it's the nitrogenase enzyme complex. So the nitrogenase enzyme complex cannot work in presence of oxygen. It is irreversibly inactivated in presence of oxygen. They cannot fix nitrogen when they are exposed to air. So rhizobial cells cannot fix nitrogen when exposed to atmospheric air. So say sentence 3 is correct. Last, the rhizobial cells get transformed into bacteroids when grown in liquid medium. Again, this ability only is present when in association with the roots of the plants. When they are in a symbiotic association with roots of their host plants, that is when the bacterial cells can differentiate into bacteroids, not when they are grown in the liquid medium. So 4 is also not correct. So out of the 4, the only correct statement is option 3, which is that the rhizobial cells cannot fix nitrogen when exposed to atmospheric air. So in that scale up system, you have to make sure that somehow the oxygen should be removed, or anaerobic conditions could be provided, or very low levels of oxygen to be maintained. Yes. Now the same explanation is given here. You can read through it later. Okay, next question. This is also related to photosynthesis in plants. Now, sucrose phosphate synthase is a key enzyme in the biosynthesis of sucrose in plants, and following are some statements given regarding this enzyme SPS. Now, which one of the following options represents combination of all correct statements? 5 statements are given. You have to identify the correct statements. First one, uridyl diphosphate glucose and fructose 6-phosphate are the substrates for SPS. Now look at the name of the enzyme, sucrose phosphate synthase. What does it synthesize? What does the name tell you? Sucrose phosphate synthase. What does this synthesize? It synthesizes sucrose phosphate, not sucrose. This enzyme, sucrose phosphate synthase, synthesizes sucrose phosphate, not sucrose. So the reaction goes something like this. See, here is sucrose phosphate synthase. This is our enzyme that we are talking about, SPS. The reaction that it catalyzes is UDP glucose plus fructose 6-phosphate. These two are added together. UDP glucose and fructose 6-phosphate are added together. UDP is removed. UDP part gets removed and glucose and fructose 6-phosphate are added together to form sucrose 6-phosphate. Okay, so this sucrose, you can see UDP is going away. Sucrose 6-phosphate is formed, and after that another enzyme comes, sucrose phosphate phosphatase. So this phosphatase will come, it will remove this phosphate group, and that's how you get sucrose. Right? So the first sentence is correct. Uridyl diphosphate glucose and fructose 6-phosphate, these are the substrates for SPS, sucrose phosphate synthase. So A statement is correct. SPS directly converts its substrate into sucrose. Does it do that? Is the product of SPS sucrose? No, right? That's what I've been telling you. The product created by this enzyme, generated by this enzyme, is not sucrose. It is sucrose phosphate. Then another enzyme, phosphatase, comes. It will remove or it will cleave that phosphate, and then you will get sucrose. So statement B is not correct. Next, phosphorylation activates while dephosphorylation inactivates SPS. This is not true. It's in fact the opposite. The statement C is opposite of actually what happens. Phosphorylation inactivates SPS, whereas dephosphorylation activates it. See here, coming back to this image, see here, when it has the phosphate, it is phosphorylated. When this enzyme is in the phosphorylated state, it is inactive. And if that phosphate is removed, then it becomes active. Right? So, statement C is not correct because phosphorylation inactivates while dephosphorylation activates SPS. That is correct. Okay, now D. So if B and C are wrong, that means D and E are correct. So what is statement D? SPS converts its substrate into sucrose 6-phosphate. That's correct. which is then converted to sucrose by the action of sucrose phosphate phosphatase. That is also correct. We just now saw that phosphatase will come, it will remove this phosphate and you will get sucrose. Last sentence, glucose 6-phosphate activates while PI inactivates SPS. Now glucose 6-phosphate can be easily converted to fructose 6-phosphate or it can be converted to UDP glucose. So these are substrates. Glucose 6-phosphate, high levels of glucose 6-phosphate, meaning that substrates are more substrates, more conversion to the product. So glucose 6-phosphate actually promotes SPS. PI, inorganic phosphate, it inactivates, basically it suppresses the activity of SPS. Okay, PI inhibits the activity of SPS or it inactivates SPS. So last sentence is also correct. So A, D and E are the correct statements and the answer is option 4. Alright, next question. Now this question is from abscisic acid biosynthesis. Plant hormone abscisic acid. So abscisic acid plays an important role in plant response to water stress. In the table below, column X represents some of the important enzymes in abscisic acid biosynthesis degradation, while column Y summarizes the functions of those enzymes. So it's a simple match the column. Okay. Now, first enzyme is 9-cis epoxycarotenoid dioxygenase, which is NCED. Now, if you've studied ABA biosynthesis, you would know that this enzyme NCED is an important step in ABA biosynthesis and it leads to production of xanthoxin. So, A goes with 3. Now, if this you identify, you should be able to very easily figure out, because this is the only option where A has already been matched with 3. There are no other options where A is matched with 3. So if this you identify, then very easily you can identify its answer or option 2. But let's go through the other enzymes. B, cytochrome P450 monooxygenase. Now this enzyme is involved in oxidation process. And as given here in option 1, it is involved in the oxidative pathway of ABA catabolism. It is involved in oxidation of abscisic acid, thereby breaking it down further. Okay, so B is 1. Then C, ABA glucosyl transferase. What does the name tell you? Glucosyl transferase. It's going to transfer glucose or is going to add glucose. And where is that option? It is option number 2. Production of a sugar conjugate form of abscisic acid. So ABA is conjugated with glucose and that enzyme is ABA glucosyl transferase. So C is 2. And lastly, beta glucosidase. So as the name says, it's going to cleave sugar. So it releases abscisic acid from its sugar conjugated form. So D is 4. Alright. So the answer is option 2. Yes. So now we are going to the evening session questions. First, let us look at the part B questions. Which one of the following is not an example of programmed cell death in plants? First, parenchyma formation in cortical root cells. There is death of cells. The cortex cells, some of them will die and that will be because of programmed cell death. So, one happens. Okay, it is an example of programmed cell death. Even second one, suspensor cell degeneration. The name itself tells you that in the embryonic state, the suspensor cells degenerate. They are induced to go into programmed cell death. It is an example of programmed cell death. Even third case, tracheary element formation in the vasculature or vascular bundle. It is basically needed for making the xylem vessels and it involves programmed cell death. But the last one, which is Casparian strip formation in the endodermis of the root. Casparian strip, as I have discussed multiple times in the classes, Casparian strip is deposition of suberin. Casparian strip is nothing but deposition of hydrophobic substances like suberin. It does not involve programmed cell death. So this is the correct answer here. It is not an example of programmed cell death. Next, which of the following is a likely consequence of a loss of function mutation in the gene which codes for the enzyme phenylalanine ammonialase in coffee plants? Now this question is from the topic of secondary metabolites in plants. Okay, now the enzyme phenylalanine ammonialase. This enzyme, you must have studied this in the second part of this topic of secondary metabolites, which is what? Phenylalanine derived compounds, phenylpropanoids or phenylalanine derived compounds. Correct? So the starting amino acid is phenylalanine, and it is acted upon by this enzyme which is phenylalanine ammonialase. The name of the enzyme tells you that from phenylalanine, ammonia is going to be removed. The NH2 amino group will be removed as ammonia. So what remains then? From phenylalanine, if the ammonia part is removed, you get an acid. Only the acid part remains, which is the cinnamic acid. So phenylalanine, after being acted upon by phenylalanine ammonialase, becomes cinnamic acid, and this pathway leads to, normally this pathway leads to the formation of lignins. It leads to the formation of lignins. So if there is a mutation in the gene which codes for the enzyme phenylalanine ammonialase, then in that case there will be a decrease in the lignins in the cell wall. There will be a decrease in the lignin deposition in the cell wall, because you have either knocked out or generated a loss of function mutation in the gene which codes for this enzyme. So this enzyme is involved in development of lignins and flavonoids and the phenylpropanoid pathway. So because of that, there is a decrease in the amount of lignins. Now caffeine, the other two options are caffeine, right? Increase in caffeine or decrease in caffeine. Caffeine is not, it's not generated because of this enzyme. The main biosynthetic pathway for caffeine starts from xanthosine. Okay, and then multiple intermediates and then caffeine. So this caffeine production is not impacted if there is a mutation in the PAL gene, which is phenylalanine ammonialase, but lignins are affected. Alright? Yes. Next question. How many molecules of acetyl-CoA condense to produce isopentenyl diphosphate, the precursor for the formation of terpenoids by the mevalonate pathway? So this question is again from secondary metabolites, the same topic as the earlier lignin question. But here we are talking about terpenoids, not phenylpropanoids. Terpenoids are the biosynthetic pathway for terpenoids is the mevalonate pathway. And this is something that you should know, right? I mean, it's not something that you can deduce. You should know that in the mevalonate pathway, this here is the mevalonate pathway. You need 3 molecules of acetyl-CoA. 3 molecules of acetyl-CoA will first produce HMG-CoA, and then HMG-CoA will be converted to mevalonic acid. That's how the pathway gets its name, mevalonate pathway. And then from mevalonic acid, isopentenyl diphosphate is generated. Okay, so the answer is option 2, that is 3. 3 molecules of acetyl-CoA are condensed to produce isopentenyl diphosphate. Next question, this is from photosynthesis. Which one of the following is the strongest oxidizing agent produced during photosynthesis? Now, strongest oxidizing agent, definitely not NADPH and definitely not ferredoxin. If you will get confused, you may get confused between these two. P680 plus and P700 plus, meaning those forms of P680 and P700 which got excited and lost an electron. Both of them, P680 plus and P700 plus, means both of them have lost an electron. And now they need to get their electron back. Now, which one is the strongest oxidizing agent? The answer is P680 plus. P680 plus is one of the strongest oxidizing agents that is biologically produced. Why? Firstly, why is it considered an oxidizing agent? Because it's going to make someone else lose an electron. How does P680 plus get its electron back? P680 plus gets its electron back by splitting water. It gets the electron from water. So it splits water molecule and takes the electron, and that's how it comes back to the ground state ready for another excitation cycle. P700 also does that, but P700 does not have that strong oxidizing capability. P680 is one of the strongest oxidizing agents produced biologically. So the answer is option 2, P680 plus. Okay, its estimated redox potential is roughly around 1.3 volts. It's one of the strongest biological oxidizing agents known. It recovers its electron by oxidizing water via the oxygen evolving complex. Now let's go to the part C questions of the evening session. First one. Now this is C4 pathway of photosynthesis. Following are some statements regarding NADP malic enzyme type of C4 photosynthesis. 5 statements are given. Which one of the following options represents the combination of all correct statements? First statement is malate synthesized from oxaloacetate in the mesophyll cells is transported to the bundle sheath cells. Yes, we already discussed this earlier that in the C4 pathway, first, PEP carboxylase converts the bicarbonate ion to oxaloacetate. Oxaloacetate is a 4-carbon compound. That's why the name of the pathway is C4 pathway. Oxaloacetate is converted to malate in the outer mesophyll cells itself. Malate is then transported to the inner bundle sheath cells. So, statement A is correct. What about statement B? What happens once the malate comes inside into the bundle sheath cells? It undergoes oxidative decarboxylation. And it forms pyruvate. So pyruvate is formed in the bundle sheath cells. Once pyruvate is formed, it is transported outside to the outer mesophyll cells where it is converted to phosphoenol pyruvate, and now it's ready for another cycle of accepting carbon. So statement B is also correct. Now the remaining 3 statements which involves aspartate and alanine, it is not carried out by this enzyme, NADP malic enzyme. NADP malic enzyme is not involved here. Aspartate synthesized from oxaloacetate in the outer mesophyll cells is transported to the bundle sheath cells and again gets converted to oxaloacetate, not triggered by NADP malic enzyme. Alanine aminotransferase converts pyruvate to alanine, not triggered by NADP malate. Here also aspartate aminotransferase is there, nothing to do with NADP malic enzyme. So, the remaining 3 sentences are not applicable at all to NADP malic enzyme. So, you can see here, this is the decarboxylating enzyme, NADP malic enzyme. So here the C4 acid which is transported from outer mesophyll to inner bundle sheath is malate, and the C3 acid which is moved from bundle sheath to outer mesophyll is pyruvate, right? So that was what statement A and B was. So A and B statements are correct. Now aspartate entering into the bundle sheath, alanine or pyruvate being formed or PEP being formed, that is not carried out by NADP malic enzyme. The enzymes are different. It is NAD malic enzyme or PEP carboxykinase. Right? So the remaining three sentences are not applicable. So the answer is option 1, that is only A and B sentences statements are correct. Next, DELA proteins are known to interact with phytochrome interacting factors and regulate genes involved in etiolation in Arabidopsis. Following are certain statements regarding the function of DELAs under dark and light conditions. So we are now going to see interaction of gibberellin signaling pathway in dark and in light. Now 4 sentences are given and you want to find out which one of the following options is the correct combination of statements. So let's look at the statements one by one. Let's look at the diagram and you will understand whether each sentence is correct or not, it's true or not. The first one, in dark, high levels of gibberellic acid helps DELA to directly bind to PIFs. This is darkness. This is what happens in darkness. The sentence says that high levels of gibberellic acid causes DELA to bind to PIFs. Now that this sentence is not true, because in darkness high levels of gibberellic acid induce degradation of DELA. Can you see that? It's written over there. DELAs are tagged for degradation via the ubiquitin proteasome pathway. So PIFs are not bound to DELA. PIFs are now free to promote scotomorphogenesis or development of the plant, morphological changes in the plant that happen during darkness. So PIF target genes are activated because PIFs are free. PIFs are not bound by DELA. So cell elongation related gene expression or etiolation related gene expressions are going on. Etiolation you understand, right? Dark grown seedlings. So dark grown seedlings, their morphological changes will continue because this is darkness. So first statement is incorrect. In dark, high levels of gibberellic acid, this part is correct. But DELA directly binding to PIF, that is not correct. Now second, during light, the levels of GA go down and helps DELA-PIF complex to bind to the promoters of the etiolation responsive genes. Now what is wrong with this sentence? In presence of light, levels of GA go down. That is true because GA is metabolized. Okay, that part is true because GA metabolism is triggered. So GA levels go down, but the second part of the sentence is not true. Okay, second part of sentence is not true. DELAs bind to PIFs. They do not allow PIFs to induce the expression of the etiolation genes. But photomorphogenesis related gene expression starts. So that's why you see the changes in presence of light. Now etiolation is not there. You don't see etiolation morphology. Now you see photomorphology. That means the changes that happen when in presence of light. So PIFs and DELAs go together. DELA binds to the PIF. So what is wrong with statement B? In presence of light, the level of GA goes down and helps DELA-PIF complex to bind to the promoters of the etiolation responsive genes. That is not true. DELA-PIF bind to each other. Yes, DELA binds to PIF. But by doing that, it is not allowing the etiolation responsive genes to be expressed. So A is not true. B is not true. What about C? binding of DELA proteins to PIF prevents the transcription of PIF-induced genes leading to photomorphogenesis. Yes, this is exactly what we discussed just now. Binding of DELA to PIFs, it prevents the expression of etiolation genes but allows the expression of photomorphogenesis related genes. So this is true. And lastly, scotomorphogenesis, morphological changes that happen in darkness, is due to the degradation of DELA proteins and binding of PIF to the etiolation responsive genes. That is also correct. So here in darkness, why does scotomorphogenesis occur? Because DELA has been degraded, it is no longer binding to PIF, and PIFs are now free to allow the expression of etiolation genes. Not photomorphogenesis genes, but etiolation genes are allowed to be expressed. So statement D is also correct. So the answer is option 4. Both C and D are true. Next, this is related to biotic responses in plants. Okay, biotic stress topic. Salicylic acid, it's a plant hormone. It regulates hypersensitive response and effector triggered immunity at the primary infection site and systemic acquired resistance in the distal tissues of the plants. Now, which one of the following statements regarding the functionality of NPR1, non-expressor of PR genes 1 in the distal tissue is correct? Again, it's not a combination of statements. 4 sentences are given. You have to find out which of those 4 is correct or true. Now, to understand this, we do need to look at the signaling pathway for salicylic acid. Okay, so let's look at it sentence by sentence. NPR1 exists as oligomers in the nucleus and activates hypersensitive response. Now, what is wrong with this sentence? NPR1 does not exist as oligomers in the nucleus. See, this is the cytoplasm. Here is the nucleus. NPR1 exists as an oligomer in the cytoplasm, and when it is activated, it is in the monomeric form, not an oligomeric form. Okay, so because of SA signaling, because of salicylic acid signaling, NPR1 monomers accumulate in the nucleus. So first sentence is not correct. First sentence says that it exists as oligomers in the nucleus. It's not correct. It exists as monomers in the nucleus. Second, NPR1 degrades through its binding to NPR3 and leads to activation of CSIR response. This is not true. In fact, even sentence 4 for that matter. Binding with NPR4 stabilizes NPR1 in the nucleus, which in turn activates the hypersensitive response. This is not true. Why? Because both NPR3 and NPR4 normally bind to NPR1 and they block the CSIR genes, they block the CSIR response. But in presence of salicylic acid, after signaling initiated by salicylic acid, NPR3 and 4 are removed, because of which the suppression of CSIR genes is now removed. Now the CSIR genes will be allowed to be expressed. CSIR genes are induced. So normally NPR3 and 4 suppress the expression of CSIR genes, but binding of salicylic acid to NPR3 or 4, it removes the suppression over the expression of the CSIR genes. So both 2 and 4 are not correct. What about sentence 3? That is correct. NPR1 accumulates in the nucleus and leads to activation of CSIR response. This is the only correct statement. You can see here, outside in the cytoplasm, NPR is a monomer, but because of SA signaling, that can happen by applying exogenous salicylic acid or because of a pathogen. By both these mechanisms, salicylic acid amounts increase. You can see NPR is now depolymerized and monomers are brought inside the nucleus, and it triggers the expression of the CSIR gene. CSIR is induced. So statement 3 is correct. The answer is option 3. Next question. Plants perceive effector molecules of a pathogen and mount a series of events that lead to activation of defense response. Following statements are made with respect to events that occur within a few minutes of the effector perception. Again, related to biotic stresses in plants. 4 statements are given. Which one of the following option represents combination of all correct statements? Now let's look at it one by one. The first statement. Because of presence of a pathogen, there is activation of defense response. Now, out of these 4 sentences, which actually occur? Okay, which of these occur as one of the defense responses? Let us see that. First, transient change in the ion permeability of plasma membrane. Yes, this happens. There is a gradual change in the permeability of the plasma membrane. So now it will allow passages of certain solutes, ions that it was not allowing earlier under normal conditions. So yes, A happens. B, efflux of potassium and chloride ions from the cell. Yes, this also happens. Plants carry out an efflux. From plant cells, there is an efflux of potassium and chloride ions from the cell. Now, what does this do? If ions are being taken out from the cell, it creates a hypertonic environment inside in the cell, and that is associated with decrease in turgor pressure. This decrease in, when potassium and chloride ions leave, that is followed by exit of water also. If water goes out, then there is a decrease in turgor pressure of the plant cell, and this decrease in turgor pressure is actually a signal. The plant is trying to send a signal to activate the defense mechanism. So statement B is correct. Statement C is also correct. On one hand, it is allowing the efflux of potassium and chloride ions. On the other hand, it allows the influx of calcium and protons into the cell. Now, if you studied this, the stress physiology in plants, that is the name of the topic. If you studied this topic, stress physiology in plants, you would know that calcium is one of the main signaling components in all the signaling pathways. Right? And because it's a main signaling intermediate, it's a signaling molecule, calcium concentration should increase inside the cell. So, influx of calcium is allowed. Why protons? Protons cause acidification of the cytosol, and that is also a defense signal. That is also a signal. Just like how increase in calcium is a signal. So, acidification of the cytosol is also a signal which activates the plant cell's defense signaling mechanism. So, A is correct, B is correct, C is correct. D is not correct because it's exactly opposite. D is saying that there is influx of potassium and chloride and efflux of calcium and proton. It's exactly opposite to B and C. So D is not correct. The answer is option 1, which is A, B and C. These 3 sentences are correct. Next question. The steady state level of a plant metabolite M is determined by the complex interplay of its biosynthesis, catabolism and transport processes from the source to the sink. Again, the same concept of source and sink has come, right? So, there is a plant metabolite M, and the question is saying that in a plant, the steady state level of that particular metabolite is dependent on which processes? Is dependent on biosynthesis of M, how it is synthesized and at what rate it is synthesized, catabolism of M, so at the rate at which it is broken down, and transport processes, the rate at which it is transported from source to sink. Now, a researcher tested the following molecular and genetic strategies for engineering the metabolite M in the native host plant. These 4 approaches are given. Okay, the 4 approaches that the researcher is testing or is going to test. The question is, which of the above mentioned strategies will provide a higher accumulation of target metabolite M in the zinc organ? Now this is important, okay? This sentence is important. You have to go through the sentences and find out which of those 4 strategies will lead to higher accumulation of that metabolite M in the zinc organ, not the source organ. You understand what source and sink, right? We discussed that earlier. So which of them will give a higher accumulation of the metabolite in the sink? So let's look at the sentences one by one. The first one is increasing the catalytic efficiency of its rate limiting biosynthetic enzyme in the source organ. Now read the sentence carefully. Increasing catalytic efficiency of what? Rate limiting enzyme, rate limiting biosynthetic enzyme. That means in the process of biosynthesis of M, of this metabolite M, there is one enzyme which is a rate limiting enzyme. And usually how do you define a rate limiting enzyme or a rate limiting step? It's a slow process. That enzyme is a slow enzyme. Its catalytic activity is super duper slow. So the researcher, if the researcher increases the catalytic efficiency of that enzyme, in very simple terms, if the researcher makes that enzyme fast, what will happen? Will it increase the rate of biosynthesis of M at the source? Yes. Yes, it will. Biosynthesis rate will increase at the source. And at the source, if more and more metabolites are being produced, then they will be easily transported to the sink. And yes, this will lead to higher accumulation of the target metabolite in the sink. Okay, so the first one is contributing to higher accumulation of target metabolite in the sink. Now second one, increasing the catalytic efficiency of catabolic enzymes of that metabolite in the source organ. Now this metabolite M in the source organ, if the catabolic enzymes are made more efficient, catabolism means what? M will be broken down. If you are doing this at the source itself, if you are making the catabolic enzymes more efficient at the source itself, will you have higher levels of M at the source? No, right? As soon as they are biosynthesized, they will be broken down. If metabolite levels are low at the source, then automatically they will be low at the sink. So B is not a strategy for higher accumulation of metabolites in the sink. Third, generating knockout of the transporter protein in the source organ. Now, at the source, on the source cells, there are these transporter proteins which are allowing the exit of the metabolite into the vasculature, into the vascular bundle, and that's how it is carried to the sink. If these transporter proteins are knocked out, if these transporter proteins which are on the source cells, if they are knocked out, can the metabolite come out into the vascular bundle? No, right? If they cannot come out of the source, how can they go to the sink? So even the third sentence or statement C does not lead to higher accumulation of metabolite M in the sink. And what about the last one? Repression of catabolic enzymes in the sink organ. So now in the sink region, if the breakdown enzymes of metabolite M are suppressed, at the sink, not at the source, at the sink, if the catabolic enzymes which break down that metabolite are suppressed, then M will accumulate. There will be higher accumulation of target M in the sink organ. So A and D, they are contributing to higher accumulation of target metabolite M in the sink organ, and that is why answer is option 4. Alright? Yes. So with this we have come to the end of this session, and Again, I hope you have understood the questions that we solved today. I hope you have a general idea now of what kind of questions to be expected. If, of course, not the same topic, but what kind of questions come and where should you put in your efforts. Again, before leaving, I would like to remind you that our enrollment for December 2024 and June 2025 exam is ongoing. You have live classes, you have recordings, you have tests, you have mind maps, you have self-study material, you have 24/7 access to chat for doubt solving. So the number is given on the slide. If you are interested, please contact this number and we are always there to help you. Alright? Even if you do not enroll, make sure that you study well for your exams, and good luck to all of you. Thank you so much. Bye-bye.
Good evening, everyone. Myself, I am Dr. Violet, and today we are here to discuss about the questions from CSIR Net June 2024. So the questions I'm going to discuss, so they are from Unit 7, that is Animal Physiology. So before I start with the discussion, please let me know if I am loud and clear and if the slides are visible. Okay. Thank you, students, for confirming. So if we go through, so first, as I've said before as well, so first we will be discussing the questions from morning session from Unit 7, and then we'll be discussing the sessions from the evening session questions. Okay, so in that also, first I'll be discussing the part B and then part C. So same, so this is for morning session. Same we will be doing for evening session. So first there are part B questions and then we are going to discuss the part C questions. So let us start with the morning session. So as we are solving the questions, you'll also come to know from which subunits mostly the questions have been asked. So the first question number is 703543. It's a part B question. It says, how many amino acids are present in calcitonin? It's a calcium lowering hormone synthesized from C cells of human thyroid gland. Okay. Now this is a factual question. So many times I have told the students that sometimes we also need to know about the chemistry of the various hormones. Okay. So if you have studied thoroughly, so there is a table, okay, that I have already shared in the classes as well, where a little bit about the chemistry of the hormones should also be known, their structure, okay, whether they have one polypeptide or two polypeptide. So some idea you should have about the various, apart from the functions, their chemistry or structure also should be known. Now according to this question, okay, as students have already started to answer. Now the answer for this question is option 2. Okay, so how many amino acids are present? So it is 32 amino acids. So it is a peptide hormone. So this is again peptide, or it is a steroid hormone. So that also should be known. And it consists of 32 amino acids. So these are synthesized by the parafollicular cells. So we know in thyroid gland there are two types of cells, right? Follicular cells and parafollicular cells. So follicular cells are basically involved in the T3, T4 synthesis, thyroid hormones. Parafollicular cells, they are involved in this calcitonin synthesis. And here if you see the functioning of how calcitonin works, so basically it will work whenever there is any disturbance or the homeostasis of the body disturbed, okay, the blood calcium levels are not optimum. Right? So suppose the blood calcium level is high, so that time the thyroid gland will release the calcitonin hormone. So if calcium level rises above the set point, calcitonin is released. So calcitonin will remove the calcium from the blood. Okay, so more calcium will now go into the bone extracellular matrix. So as a result, blood calcium level will fall and the body will return back to homeostasis. Okay, and vice versa. Suppose the calcium level is less in the body. Okay, so level falls below the set point. At that time, instead of releasing calcitonin, the parathyroid gland, okay, it is another gland that is present on top of the thyroid. So here on top, as you can see, there are parathyroid glands. So they release this parathyroid hormone. Okay, now this parathyroid hormone, what it does, it works just the opposite of this calcitonin. So it will do bone resorption, where this bones they'll give off. So as a result, the calcium levels will increase in the blood. Okay, so blood calcium level rises when parathyroid hormone is released. So calcitonin and parathyroid hormone, so they work opposite to each other, and in this way they regulate the blood calcium levels. So according to this question, calcitonin has 32 amino acids. Now moving on to the next question, 703544. The retinal rod cell cyclic GMP phosphodiesterase is an enzyme with subunit structure. Now we know the functioning of phosphodiesterase, what it does, but here again you can see that a little bit you should have idea about the structure of this enzyme. Okay, now so some students have already answered. Yes, the correct answer over here is option 2, that is alpha beta gamma 2. So the cyclic GMP phosphodiesterase is an enzyme with the subunit structure alpha, beta and gamma. Okay, so it has these 3 polypeptides. So this enzyme is crucial, so it helps in phototransduction pathway, right, where it hydrolyzes cyclic GMP to GMP. So in this way, it contributes to regulate the visual signal transduction in rod cells of retina. So basically it helps in vision. Okay. So it is a key effector enzyme of vertebrate photoreceptor cells, and it regulates the level of the second messenger, that is cyclic GMP. Okay, so it converts cyclic GMP to, or you can say it hydrolyzes cyclic GMP to GMP. Now this enzyme, this phosphodiesterase, consists of alpha and beta subunits, and this gamma is actually a inhibitory subunit. Okay, so it has 2 inhibitory gamma subunits. So it will block this phosphodiesterase activity in the dark. Okay, so accordingly the right answer will be alpha, beta, and there are 2 gammas. Okay. So the right answer is option 2. Now let's move on to the next question. It's the question number 703545. Now again it is a factual question. Okay, it is from sensory organs. How many times a sound wave is amplified in the middle ear of a human? Okay, so we know ears are divided into 3 regions. Okay, outer ear, middle ear and inner ear. So the inner ear is the most important part of the ear, while the middle ear, it plays a role in amplifying the sound waves, right? Where we have the 3 bones and the vibrations get passed and they get amplified. So the question is, how many times the sound waves get amplified? So for this you need to know, okay, how many times the amplification occurs. So the right answer over here is option 1. Okay, we will take the range 16 to 18 times the sound waves are amplified. So the vibrations, so as you can see the figure over here, this is the outer ear. So sound waves are collected by the pinna and it passes the auditory canal, hits the tympanic membrane, and then in the middle ear we have this malleus, incus and stapes in the oval window, as well as round window is a part of the middle ear. And then this is the inner ear where the vibrations will pass through perilymph and endolymph in the cochlear duct. So here, so when these vibrations are passing, Okay, so the vibrations of the tympanic membrane, so when it hits this tympanic membrane, it gets amplified by around 20 times, okay, as the energy is passed to the foot plate of the stapes. So the stapes is in the oval window, so the sound waves pass to the foot plate where it impinges on the membrane of the oval window. So how many times it is getting amplified? It is around 20 times. Although it is 20, but here you don't see 20, but the most correct option, okay, nearest to 20 will be 16 to 18. Okay, so that is why the right answer for this question is option 1. Next question is from nervous system. Okay, so again nervous system is important, we know. So if you see the questions from part B, so one is nervous system, sensory organs, then this is also we can consider from sensory organs. Okay, and this is from hormones. So as expected, the questions are from the important subunits like endocrinology over here, then from sensory organs, and then nervous system. So here the question is, which one of the following neurotransmitters is synthesized in synaptic vesicles instead of being transported to the vesicle after its synthesis in the cytoplasm? So usually what happens, the neurotransmitters, they are synthesized in the cytoplasm, okay, and whenever and then they will be transported to the vesicle. So that is how the neurotransmitters enter the vesicle, okay, so that is the usual way. But here there is one exception where the neurotransmitter is itself synthesized in the synaptic vesicle instead of coming from the cytoplasm directly. So for this question, definitely we need to know about the pathways actually that helps in the synthesis of various neurotransmitters. Okay, then only we'll be able to solve this question. Okay, can anybody tell me the right answer? Okay. Okay. So the right answer for this question is option 1. Okay. So norepinephrine is actually synthesized in the synaptic vesicles. Okay, but it is not that completely, from scratches it is synthesized. We'll see how it is being synthesized in the synaptic vesicles. So there are two main categories of neurotransmitters: small molecule transmitters and peptide transmitters. So synthesis and storage of these neurotransmitter groups also differ. So most small molecule transporters, they are synthesized by enzymes that are present in the cytoplasm, except for norepinephrine. So neurons that release norepinephrine, which is another catecholamine transmitter. So once dopamine is packed into the synaptic vesicles, a membrane-bound enzyme called dopamine beta hydroxylase will convert this dopamine into norepinephrine. Therefore, unlike the other small molecule neurotransmitters, norepinephrine is synthesized within the vesicles, not in the cytoplasm. Okay, so if you see, like how in the cytoplasm, till here dopamine is synthesized. Okay, so tyrosine, it is transported into the adrenal chromaffin cell, okay, with the help of various enzymes. So it first formed dopa and then dopamine is formed. So this dopa is the decarboxylase. Okay, and here this DD is dopa decarboxylase enzyme. Okay. So dopamine will enter into the vesicles, and inside the vesicles there is DBA, that is dopamine beta hydroxylase enzyme. Okay, as I just told you all over here. So this dopamine beta hydroxylase will convert this dopamine into norepinephrine. Okay, and further the norepinephrine will go outside into the cytoplasm and further changes occur. That is how it forms the epinephrine. Okay, but norepinephrine, where it is being synthesized? Inside the vesicles. Clear? So that is why the right answer for this question is option 1. And others are synthesized in the cytoplasm. So that was about the part B questions. Now let's discuss the part C questions. So the first question in part C from morning session is 703607. So as we can see, the question is from circulatory system. Okay, so that is well expected. Let us see how to solve this question. The stimulation of sympathetic cardiac nerves increases the rate of action potential generation from SA node of heart. The following statement suggests the mechanism of this action. A. The depolarizing effect of H current or IH is decreased by sympathetic stimulation. B. Norepinephrine secreted by the sympathetic endings binds to beta-1 adrenoreceptors, resulting in the increase of intracellular cyclic AMP. C. The increased intracellular cyclic AMP facilitates the opening of long-lasting calcium channels. D. The calcium current due to opening of voltage-gated L calcium channels is decreased. So here we have to see the effect of what is happening when there is stimulation of sympathetic cardiac nerves. Okay, and accordingly we have to select which one of the following options represent the correct combination of the statements. Okay, yeah. Can you all try and solve this question? Okay, yes, I can see some correct answers. So right answer for this question is option 2. Okay, B and C are the correct statements over here. So first we have to consider, okay, what all happens when there is sympathetic stimulation of the autonomic nervous system. So the first statement says the depolarizing effect of IH is decreased by sympathetic stimulation. So this H current, okay, so whenever there is sympathetic stimulation, so this graph is very important, okay, not only for this exam, many times you all will see that same type of question is asked in the previous year question papers also. So already in the class we have discussed this graph. So this is sympathetic stimulation, so this is the normal graph under normal conditions. When there is sympathetic stimulation, the curve is towards the left, and when there is parasympathetic stimulation, the curve is towards the right. So if the curve is towards the left when there is sympathetic stimulation, so you can see what all changes occur. Okay, so the phase 4 slope will increase. Okay, while if it is parasympathetic, the slope decreases. Okay, and other changes accordingly will occur. So when there is sympathetic stimulation, norepinephrine is released, binds to certain receptors, that is the beta adrenoreceptors. So it increases the funny pacemaker currents. Okay, so the current will increase. So IH will increase instead of decreasing. The IH is written decrease, so this is incorrect. Okay, so when there is a depolarizing effect, the currents will increase. And next statement says norepinephrine secreted by sympathetic endings binds to beta-1 adrenoreceptors, resulting in increase of intracellular cyclic AMP. So yes, as I said, okay, so they will bind to beta adrenoreceptors and there will be increase invert calcium current. So calcium currents will increase. So that is why depolarizing is occurring. So again, B is correct over here. Okay, and what is C? Let us see. The C statement says the increased intracellular cyclic AMP facilitates the opening of long-lasting calcium channels. So yes, because norepinephrine is entering, cyclic AMP will increase, and the increase in cyclic AMP will further help in the opening of L calcium channels, because first the slow sodium channels open, IF currents are generated, and then what happens, the T-type calcium channels open, then the L-type calcium channels open. So during this, when the cyclic AMP increases, it will further help in the opening of long-lasting calcium channels. Okay, so D, so in this case again C is correct, and the GES subunit of the beta 1 adrenoreceptor, when it gets activated, it upregulates the enzyme. Okay, so when it binds to these receptors, adenylate cyclase increases, so it convert the ATP to cyclic AMP. Okay, so that is why we mentioned that intracellular cyclic AMP levels will increase. Okay, so B and C is correct over here. Let us talk about D. D says the calcium current due to opening of voltage gated L calcium channels is decreased. So no, as we said, it will further enhance the opening of L type calcium channels. So that is why D is again incorrect. So the right and the correct answer for this question is option 2. B and C statements are correct over here. Let's move on to the next question. So this question, we can say it is from respiratory system. 703608. So during physical exercise, a large amount of oxygen is delivered to the active muscles by many physiological adjustments, including a change in the the P50 value, which is determined by the partial pressure of oxygen at which hemoglobin is half saturated with oxygen. Okay, so that is the P50 value when at a particular partial pressure of oxygen when the hemoglobin is half saturated with oxygen. So the following proposed statements explain the mechanism of change in this P50 during exercise. A. P50 is increased during exercise and the temperature rises in active muscles. B. During exercise, metabolites accumulate in the active muscles, resulting in higher pH that increases P50. C. P50 is increased during exercise as CO2 is decreased in active muscles. D. An increase in 2,3-DPG has been reported in non-trained person within 60 minutes of exercise, resulting in higher P50. So according to this question, okay, so you all can try and tell me, so which of the following options represent the correct combination? So we have to find the correct statements over here. Okay, so we'll try to solve this question. Okay, so for this question we need to know what all is happening whenever a person is exercising. Okay, whether temperature increases, pH decreases, okay, and what effect it will have on the P50 value. So the right answer for this question, it is option 4. Okay, A and D statements are correct over here. So whenever there is an increase in tissue metabolism, that is when the person is exercising, so it will increase in local temperature, so temperature will increase. The hydrogen ion levels will increase. Okay, so that is why there will be acidic conditions. And partial pressure of CO2, okay, so that will also increase because the person is exercising, right? CO2 partial pressure will increase. So all of which, what it will do, it will increase the P50 value. So that is why when P50 is increasing, so A is correct and B and C are incorrect statements over here. So if we see, P50 is increased during exercise, the temperature rises, so this is correct. B, during exercise, metabolites accumulate, resulting in higher pH. No, the pH decreases, right? So that is why so this statement is incorrect. pH does not increase when there is exercise. Okay. And P50 is increased during exercise as CO2 is decreased in active muscles. So whenever a person is exercising, CO2 will increase, it will not decrease. So that will increase the P50. Okay, so again C is incorrect. And the metabolites are also getting accumulated. Okay, so that will result in higher P50 values. So D is again correct over here. Okay, so that is why A and D are the correct statements. So the right answer is option 4 over here. So if you see this figure, okay, and this is an important figure. The graph is very important. Okay, many times you will see that the same type of questions are asked in various CSIR question papers, just that the statements might little bit vary. So the reaction over here is oxygenated hemoglobin. Okay, so because H plus is high when there is acidic condition, so this H plus can replace the oxygen. So that is how oxygen is released to the cells. Okay, so this is the normal graph so it's a graph plotted on the y-axis with percent saturation of hemoglobin, and on the x-axis it is plotted with respect to the partial pressure of oxygen. So the curve will move towards right whenever partial pressure of CO2 increases, or there is acidic conditions where H plus will increase, or the temperature increases, or the metabolites increases. So when the curve moves towards right, affinity of hemoglobin for oxygen will decrease, as mentioned over here. And in other conditions, like if the partial pressure of CO2 decreases, or H plus decreases, pH increases, or temperature decreases, metabolites decreases. So in all these conditions, or if it is a fetal hemoglobin, the curve will move towards left. So whenever the curve moves towards left, affinity of hemoglobin for oxygen will increase. Okay, so this is something that very basic we need to know, and we need to remember these factors, how it affects this oxygen saturation curve, that this curve is known as. So let's move on. Yes, before I go to the next question, I wanted to share about the Josh batch that will be starting, and for enrolling into Josh batch, you can call on the toll-free number. So here in Josh batch, you will be well practiced for preparing for December 2024 as well as June 2025 exam. You will get access to various special supplements on general aptitude as well as practice papers, AIM test series, and you will have 24/7 chat support. Students who enroll for Josh batch, they'll get free access to Biotecnika's online study portal, free access for the Conceptual Cloud. Internship will be free, mind maps. You will have access to various mind maps who all enroll for Josh batch, as well as study material, self-study booster package is announced for this batch, where there will be AI-powered AIMNet e-test series as well, ToughNet e-question bank, concept table, concept wheel, concept cloud, and definitely it is going to give you all success. So it has double success guarantee with premium guidance via chat support and live sessions from the best teachers. So please, you all can enroll for it, who all needs to enroll for Josh batch, as well as your friends, your in your workspace, in your colleges, whoever is interested in preparing for CSIR net, do inform them about the Josh batch. Okay, now let's move on to the next question. So this question is again from nervous system. So it is not very simple, I would say, because here you need to know about it's the match the following. Okay, so you need to match the type of mammalian nerves fibers and the conduction velocity in meter per second of nerve impulses, that is in column Y. So for this you need to exactly know the values, only then you will be able to solve this question. Okay, but yes, they have asked this type of questions before as well. Okay, so that is why it's very important that we solve the previous year question papers. So let us see this question. So you can say it's like a factual question, then only you can match the values. So which of the following option represents correct match between column X and column Y? So the right answer for this question is option 3. Okay, now let us see the characteristic of each of these mammalian nerve fibers and their velocity. So the conduction velocity of A alpha is 80 to 120 meter per second. Okay, A alpha 80 to 120, so A will match with 3. Okay, for B, for A beta, it is 30 to 70 meter per second. So this is A beta, so it will be 30 to 70. So D will match with 2. Next, A delta is 12 to 30 meter per second. So this will match with 1. C will match with 1 and B will match with 4. Okay, 3 to 15. So the right answer is option 3 over here. So the correct match is option 3. So in order of decreasing size are the different nerve fiber groups. So if you see the nerve fibers, alpha, beta, delta, so this is basically their size is decreasing. That is why they have been named like that. So we have seen there are three types, right? A, B and C, and again there are subtypes in that, okay, depending on their size. So A alpha is there, A beta is there, A delta, okay. So A alpha, A beta, A delta and V nerve fibers. So the thickness of the nerve fiber is correlated to the speed with which information travels in it. So if you see, if it is more thicker, okay, so diameter is higher for A alpha, so the speed will be higher. Okay, so if you have some idea about their diameter, so you can easily solve this question. So A beta, diameter is less, so speed is less. A delta is still less, so alpha, beta, delta. So in this way, their size decreases, so their speed, conduction velocity will also decrease. Okay, and C is the diameter is very less, so 0.2 to 1.5 micron, that is very less. So speed is also very less, in points it is. Okay, but according to the question, if you see, so A alpha, then beta. So alpha has 70 to 120, then beta 12 to 30. Okay, and delta, A alpha, A beta is 30 to 70, A delta is still less, 12 to 30, and B will be lesser than A. Okay, so if you know this, this fact that the speed depends on the diameter, then you can easily solve this question. Okay, so B will match with 4, where the B diameter will be less compared to A. Right, and again C is much lesser, it is in points. So here C is not given, but yes, so that is how you have to try to solve this question. So even if you don't know the exact values, if you have the understanding of the diameter, how it affects the speed, okay, then you can easily solve this question. So I would say this question is, it's not very difficult or not very easy. You need to have this idea, that is, it has medium difficulty level. But yes, if you know this, then it is very simple and you can easily solve and score 4 marks. Now let's move on to 703610. The following statements are made about the variety of thermoregulatory mechanisms in the body. A. Human voluntary activity is decreased in cold. B. There is a cutaneous vasodilation by heat. C. There is an increased secretion of epinephrine and norepinephrine in cold. D. There is a decreased heat production in cold. So choose the combination of all correct statements about thermoregulatory mechanisms. So this is from thermoregulation chapter. And we have to find the correct statements. So if you go through the previous year question papers, so definitely from thermoregulation they are asking questions. And this is something that is very general type of question. Okay, we should know how our body tries to regulate the temperature so that it does not, it is in the optimum conditions. So yes, anybody can solve this question. Okay, so let us see how to solve this. So the right answer for this question is option 2. B and C are the correct statements over here. So what it says, human voluntary activity is decreased in cold. So when the environment becomes too warm, so thermoreceptors trigger vasodilation. So this is about this statement, okay, where there is a cutaneous vasodilation by heat. So this is correct because whenever the body temperature increases, so there will be vasodilation, so that more heat goes towards the peripheral parts of the body, right? So as a result, the heat should be dissipated. Okay, it will be released to the environment. So B is correct. And this directs blood flow towards the skin where excess body heat can escape. So B is correct. During cold, heat is produced via shivering. Okay, so that is why D is incorrect. So D says there is a decreased heat production. No, whenever we feel cold, you might have realized what we do, we shiver, right, if we are very cold. So that is done to increase the heat production, not to decrease. Okay, so there will be increase in heat production in cold. So D is incorrect over here. Now let us see the other statements. Voluntary activity decrease is not a thermoregulatory mechanism. Okay, so that is why A is incorrect. So that is something voluntary, something that is depending on us, right? So it is not a thermoregulatory mechanism. Okay, so A is not accepted. It is incorrect. Then about the stress hormones that are released. So yes, when the body is in any kind of stress hormones will increase. So as a result, option C is again correct over here. Okay, epinephrine or epinephrine. So these are related to stress and they will be released. So C is correct. So the right answer is option 2. So if we see this figure, okay, what all happens, how the body tries to thermoregulate. So whenever we feel cold, okay, so there is vasoconstriction. So less blood will flow towards the peripheral parts of the body. Okay, and shivering, non-shivering. So in this way, heat is generated. So there are two ways, shivering and non-shivering thermogenesis. Okay, brown adipose tissue are involved over here in generating heat. Okay, so that is trying to regulate the body temperature when it is cold conditions. If it is very high temperatures, to regulate that, so there is increased core or surface temperature. So that time we undergo sweating, right? So more heat. So when we sweat, so heat is the water, the sweat. Okay, so it's basically water. So it will be evaporated. So that will give a cooling effect. So as a result, it will try to maintain the body temperature, as well as there will be vasodilation. Okay, so more blood will go towards the peripheral parts of the body so that the heat is released out. Okay, so this understanding is very important. Okay, so any question that you see from thermoregulation is based on this understanding of the concept of how thermoregulation tries to maintain our body temperature. Now let's move on to the next question. So this question is from blood and circulation chapter, that is unit 7A. And basically we know from unit 7A, hemostasis is very important. Okay, recently in other CSIR question papers also they had asked a similar question about the various blood clotting factors. But yes, now if you know the names and the numbers, then only you can solve this question correctly. Okay, but yeah, some of them we already know. Let us see how to match this and find the correct answer. So, given below are blood clotting factors in column X and their names in column Y. So, let us see the table first. Okay. So, though some of it, we already know what are the names, like D. Okay. 10 will match with 3. So, D we know is a stored power factor. Okay. And then we know pre-Ka, kallikrein. Okay, so pre-Ka will again match with Fletcher factor. Okay, and then 12. So this 12, that is A, is going to match with Lackey-Lorand factor. Okay, so if you know that, so the right answer will be option 3 over here. 703611. The right answer is option 3. And HMWK, so this is going to match with Fitzgerald factor. Okay. So how you can solve this? You can use this table, okay, where you have the various numbers and their names. So like 10 is Stewart factor, okay. Then we saw PK, pre-kallikrein, is Fletcher factor. HMWK full form is high molecular weight kininogen, okay, that is HMWK, and it is also known as Fitzgerald factor. Okay, and for 12 you have other names as well, okay, like Hageman factor, and it's a part of the intrinsic pathway. So the right answer is option 3 over here. So many times, they have asked questions like this, the names and their numbers where you have to match. So you can use this table, okay, try to remember the names, okay, and some of them, like, they have more than one names, okay, like PK, pre-kallikrein, Fletcher factors, all this you should learn it by heart. Like 3 is also tissue factor, thromboplastin. right? So that's a simple question. Now next move on to question number 703612. So it is again expected. So this question is from excretory system. Okay, so as I mentioned in the class also, like, the very important topics are circulatory system, respiratory system, nervous system, excretory system. Okay, this time from endocrinology they have not asked, but yes, that is also important. But yes, they have asked in the part B. So now let us see the excretory system question. The following statements are made for the effect of hormones on the GFR, glomerular filtration rate. A. Norepinephrine, epinephrine and endothelin constrict renal blood vessels and decrease GFR. B. Endothelin dilates renal blood vessels to increase GFR. C. Norepinephrine and endothelin constrict renal blood vessels and decrease GFR, while epinephrine dilates renal blood vessels to decrease GFR. D. Prostaglandin and bradykinin decrease renal vascular resistance and increase GFR. So many times I have asked the students to make a table between the various vasodilators and the vasoconstrictors. Okay, so if you have made that table and you have studied that before the exam, so this question becomes easy. Okay, so just you have to know whether it constricts or whether it dilates. So the right answer for this question is option 4. Okay, where A and D are the correct statements. So let us see each of these statements. So with greater sympathetic stimulation during exercise or hemorrhage, so that time the sympathetic division gets activated. So vasoconstriction of afferent arterioles predominant. So as we know, so this is afferent, this is the glomerulus, and efferent. So when there is sympathetic stimulation, so hemorrhage, blood loss, right? So the body don't want to lose water. So this blood vessel will constrict. Okay, diameter will decrease. So as a result, blood flow is decreased. Okay, so when blood flow is decreased, GFR will decrease. So that is why A is correct over here. Okay, so when there is norepinephrine, epinephrine and endothelin, so they are constricting the renal blood vessels, so that will decrease the GFR. Okay, so endothelins, these are constrictors, they will decrease the diameter, so A is correct. B, it says endothelin dilates renal blood vessels, so now only we saw it is a constrictor. Okay, it will not dilate, so B is incorrect. C, norepinephrine and endothelin constrict renal blood vessels and decrease GFR, while epinephrine dilates renal blood vessels to increase. So this is incorrect. Okay, so they work together. So C is not correct. D, prostaglandins and bradykinin decrease renal vascular resistance and increase GFR. So what they do, bradykinin is a product of the action of kallikrein on kininogen. So bradykinin induces vasodilation. Okay, so it will increase the blood flow, as well as natriuresis, means it will allow the sodium to move out. So in turn, this bradykinin, how it does that? The releasing NO and prostaglandins. So prostaglandins, the best defined renal functions for prostaglandins is PGE2 and prostacyclin. So these are again vasodilatory in nature. Okay, these vasodilatory prostaglandins increase the renal blood flow and GFR rate will increase. So in that way, D is correct over here. Okay, it increases GFR. So if GFR is increasing, diameter is increasing, so the vascular resistance will decrease. Right, resistance to the flow of blood will decrease because the diameter is increasing. Okay, so D is correct over here. So A and D are the right statements. So if you see this figure, how the GFR is being regulated. Okay, so we have two ways: intrarenal, intrinsic activity, and extrinsic, how it will try to regulate the GFR. Okay, so maintain constant GFR over a wide range, while extrinsic, like the hormones and the, so intrinsic means the kidney itself, renal autoregulation that we have studied. Okay, itself, it will play a role through myogenic response and tubuloglomerular feedback to regulate the GFR, while extrinsic mechanisms will involve the nervous system and the hormones that are involved. So through the nervous system, the sympathetic division will get activated. So it will maintain blood volume and pressure, regulate the GFR. Okay, and through the RAA mechanism where angiotensin 2 is released. So in that way also it will try to increase the GFR if the GFR is less. Okay, So that was all from the morning session. So if you go through, the questions are asked from excretory system, okay, in part C, from hemostasis mechanism, from thermoregulation, nervous system, respiratory and circulatory system. Okay, so all these are very important subtopics in unit 7. Study them thoroughly. And now let's move on to the next session. So in total, if you see the part C questions, there are around 6 part C questions. Okay, so even if you solve 4, so and you're confident of it, so you will get good scoring, right? Because they're not very difficult as we went through the various questions. If you have understanding of what are various vasodilators, various vasoconstrictors, okay, you know about the various blood clotting factors, so it's easy to solve. But yes, you should be very confident of your answers. You should not be any kind of dilemma, because otherwise in that case we tend to choose the incorrect statements. So try to be very confident when you are studying. You should know very well whether this epinephrine is acting as a vasodilator, or whether nitric oxide is a vasodilator or a vasoconstrictor. So all this should be very clear. So let's move on to the next session. So the first question, so again we are first going through the part B question. The first question number is 703044. Which one of the following hormones is not exclusively synthesized from a single location in the body? 1. Thyrotropin releasing hormone. 2. Corticotropin releasing hormone. 3. Somatostatin. 4. Somatotropin. So this is a very simple question. Even in classes we have discussed. Okay. So anybody can tell me the right answer. Okay. Yes, the right answer, correct. So the right answer for this question is somatostatin. Okay. If you have gone through the classes PowerPoint, so we have already discussed this part. So thyrotropin releasing hormone, so it is released from where? From the hypothalamus. So it is just one location from where it is synthesized, TRH. Corticotropin releasing hormone is synthesized again in the hypothalamus, but for somatostatin it is produced in many locations. So we have studied it in the pancreas, we have studied it through the hypothalamus as well. Then apart from that, from gastrointestinal tract also it is synthesized, and the central nervous system. So it is produced in many locations. So that is why the right answer is option 3. While somatotropin is synthesized in the pituitary gland. Okay. So that's 703044 done. Now let's move on to 703045. So again it is about the receptors. So which one of the following adrenoreceptor decreases cyclic AMP in the postsynaptic target after stimulation with norepinephrine? Means, so norepinephrine binds to its receptors, but here it is trying to decrease the cyclic AMP. So which adrenoreceptor is involved? For 403045, it is the alpha 2. Okay, so the answer is option 2 over here. So if you see this figure, so the alpha 2 adrenoreceptor is a G protein coupled receptor. It is a GPCR. Okay, so this is alpha 2 is associated with inhibitory, and this is, so this when stimulated by norepinephrine, this alpha 2 adrenoreceptor over here, it activates the inhibitory G protein. Okay, so this activation will decrease the cyclic AMP in the postsynaptic target cells. So through this figure also we can understand. So when it binds to the alpha 2, so inhibitory activity increases, inhibitory G protein gets activated, so that will decrease the cyclic AMP. Okay, and if it is alpha 1, then you can see IP3 will increase. but because they have asked for cyclic AMP, so it is your alpha 2. Clear? Now let's move on to question number 703046. So the question is, which one of the following gases diffuses through alveolo-capillary membrane in shortest time at the resting condition? So these are the various gases, but we have to find out which will be undergoing fastest diffusion. Okay, shortest time means faster it can diffuse. So anybody can try and tell me? Okay, yes, the right answer for this question is option 4. Okay, N2O will be the right answer. The gas that diffuses through the alveolo-capillary membrane in the shortest time at resting conditions is nitrous oxide or N2O. This is because the rate of gas diffusion across the alveolo-capillary membrane depends on the gas's solubility in the blood and its molecular weight. So accordingly, N2O has a high diffusion rate due to its low molecular weight and relatively high solubility in the blood. So it allows it to diffuse quickly across the alveolar membrane. Okay. So if we compare between oxygen and CO2, it is CO2 that diffuses faster. But if we compare between all these various gases, so the right answer is N2O over here. The next part B question, so this is from hormones, okay, related to the renal hormone. 703047. Which one of the following is considered as a renal hormone? Okay, so there are various names that are given: megalin, cubilin, renalase, uroguanylin. Okay, so it is a factual question. You need to have information, then only you can solve this question. So the right answer for this question is option 3. Okay, renalase is the correct answer. Let us see the other hormones and their characteristic, the other molecules that are involved. So renalase over here, it is a relatively recently discovered kidney protein, okay, connected to chronic kidney disease and blood pressure disorders. So what it does, it functions by metabolizing the catecholamines, that is epinephrine, norepinephrine, and similar substances such as adrenaline and dopamine. So it is a recently discovered renal protein, okay, and it is a hormone, okay, that will regulate the blood pressure disorders. Megalin is a large endocytic receptor found in kidney. So it is not a hormone, it is a receptor. It plays a role in reabsorption of various proteins, but it is not classified as a hormone. Cubilin is another receptor involved in reabsorption of proteins. So this is again a receptor and other molecules. So it can reabsorb various other molecules. Like megalin, it is not a hormone but a receptor protein. Uroguanylin, so it is a 16 amino acid peptide. It is secreted by enterochromaffin cells in the duodenum and proximal small intestine. Okay, so it is not a part of the kidney, it is not a renal hormone, uroguanylin. So the right answer over here, that is why it is renalase. Okay, so know about the other molecules as well, okay, apart from renalase. So that was all from part B. So if we go through the questions are asked from hormones, from respiratory system, okay, from the various receptors, okay, and again another hormonal question. So endocrinology, from endocrinology they have asked 2 part B questions. Now let us move on to the part C question. So the question number is 703107. It's from nervous system. Different phases of a typical nerve fiber action potential are explained in the following statements. The membrane potential is brought to the threshold potential, that is the firing level, due to the opening of some voltage gated sodium channels in response to a threshold depolarizing stimulus. The rapid depolarization after the firing level is caused by opening of more voltage gated sodium channels and entry of sodium into the nerve fiber. See, the reversal of membrane potential, that is the overshoot at the peak of action potential, occurs as membrane potential moves towards the equilibrium potential of potassium. D. The peak voltage of action potential does not reach the equilibrium potential of potassium primarily because the increase of potassium conductance is short-lived. So, which of the following options represents the combination of the correct statements over here? So, it's a simple question, simple from action potential. Okay, direct question. So, can you all tell me the right answer? Okay, yes, that's correct. So, the right answer for this question is option 1. So, it is quite obvious. So, let us understand the various statements over here. So, first statement says the membrane potential is brought to threshold potential due to opening of some voltage gated sodium channels. Definitely what happens in the graph? Okay, so first it reaches the threshold. Okay, so that time the sodium channels open. Right, so statement A is correct. Statement B says the rapid depolarization after the firing level, that is after the threshold, is caused by opening of more voltage gated sodium channels. So that is also correct because initial depolarization, some sodium channels will open and then there is fast depolarization where large number of voltage gated sodium channels open and sodium enters inside. So B is also correct. C, it says the reversal of membrane potential, okay, where after depolarization it becomes repolarization, or the overshoot at the peak of action potential occurs as membrane potential moves towards the, so it is not because it is moving towards the equilibrium of potassium, no. So first it will try to move towards the equilibrium of sodium, but it will not reach. Okay, so that is why this part of the C statement where it says towards the equilibrium of potential of potassium, that is incorrect. D, it says the peak voltage of action potential does not reach the equilibrium potential of potassium primarily because the increase of potassium conductance is short-lived. Now here, as I said, it is not the equilibrium potential of potassium. It is trying to reach the equilibrium potential of sodium. Okay, so again D is incorrect. So if you see this graph, so accordingly the right answer is A and B, that is statement or the option 1. Now let us see this graph. So you can see there is a stimulus, okay. There is depolarization, it will reach a threshold. Resting membrane potential minus 70, it will reach a threshold of minus 55 and then it will depolarize and it will try to reach towards the equilibrium potential of sodium, but it won't reach. Before that, sodium channels will close and potassium channels will open. So potassium will now move outside of the cells. As a result, it will repolarize, it will try to reach towards its resting membrane potential, and because more and more of potassium channels will be opening, so as a result, it will become more negative. So this is the phase where hyperpolarization will take place. Okay. So let us quickly see the statements. A threshold depolarizing stimulus opens some voltage gated sodium channels, causing the membrane potential to reach the threshold level, initiating the action potential. So this is correct. That is what is statement 1. And the statement A. The next statement was statement B, where now we saw that it was a correct statement. So here it was talking about rapid depolarization after the firing level is caused by opening of more voltage gated sodium channels. So once the threshold is reached, more voltage gated sodium channels open. So there is a rapid influx of sodium and causing the depolarization phase of the action potential. So that is why the statement B is correct. The overshoot occurs because the membrane potential moves towards equilibrium potential of sodium. Okay, as I said in the beginning. So this statement is incorrect. That is the statement C. The peak voltage of action potential does not reach the equilibrium potential of sodium because it is not the potassium, it is the sodium, because the sodium channels begin to inactivate and potassium channels, as I said, start to open. So that causes the repolarization phase. So that is why the D statement is again incorrect. So the correct statements over here are option A and B. That is, the statement A and B, option 1 is correct over here. So you should study about action potential generation thoroughly. You can use Gerard et al., the standard reference book, where everything in details is mentioned about how the action potentials are generated in the neurons. Next is question number 703108. So it's a simple question from excretory system, where it's just a match the following, where different segments of the renal tubule are given in column X, and the sodium transporter in the apical membrane of tubular cells are mentioned in column Y. So which of the following options represents the correct match between column X and column Y? Can you all try and tell me? I'm sure it is very simple. Okay, so some students have started to answer. Okay, yeah, so let us see the correct answer for this question first. So the correct answer is option 4. Now let us see. Proximal tubule means there will be sodium symporters, right, that are used for the reabsorption of amino acids, glucose, right? So A will match with 4. Sodium amino acid co-transporter. B, in collecting duct, definitely there are sodium channels. Okay, sodium chloride channel. So B will match with 1. C, the thick ascending limb has 1 sodium, 1 potassium, 2 chloride co-transporter. So this is very simple. C matches with 2. And again D, the DCT part of the renal tubule, you have sodium chloride co-transporter, right? Sorry. So now we see that the correct answer for this question is option 4. Right? I would say it's a very easy question. Who all might have solved would have got it correct. So as mentioned in the PCT, sodium ions are reabsorbed through the apical membranes via sodium amino acid symporters. So a match with 4. In the thick ascending limb of nephron loop, the sodium potassium 2 chloride symporters will reabsorb sodium and chloride from the tubular fluid. The early or initial part of DCT will reabsorb 10 to 15% of filtered water, 5% of filtered sodium and 5% of filtered chloride. So that is why reabsorption of sodium and chloride occurs by means of sodium chloride symporters. Okay, so the right option is the option 4. So let's move on to the next question. Now again, this question is from thermoregulation. Okay, the question number 703109. So there are some statements. Let us quickly go through them. In hot environments, some changes occur in the human body to improve heat tolerance, which is called heat acclimatization. The following suggested statements describe the physiological adjustments during heat acclimatization. Vasoconstriction starts in skin at a lower body temperature. B, salt concentration in sweat is increased. C, the sweat secretion over the skin is more effectively distributed for optimum use of the effective surface area for evaporative cooling. D, the sweat glands maintain high output for longer periods. E, the threshold for start of sweating is increased. So we have to find which of the following options represents the combination of the correct statements. Yes, you all can try and tell me. So you have to think about what all changes occur during heat acclimatization. In the morning session also we saw a similar question from thermoregulation. So definitely when heat acclimatization has to take place, there should not be vasoconstriction, right? It should be vasodilation. So A is not correct. What about B? Salt concentration in sweat is increased? No, salt concentration will not be increased, right? Because if salt concentration increases, so more and more of water loss. Okay, so dehydration might occur. So B is also incorrect. C, the sweat secretion over the skin is more effectively distributed for optimum use of the effective surface area. Yes. So this is what happens to acclimatize to the heat. Sweating occurs and it is equally, effectively distributed, so that when sweating occurs, evaporation will occur and that will give a cooling effect. So the heat of the body will be distributed and released. So C is correct. D, the sweat glands maintain high output for longer periods. Yes. So this is also a correct statement. And E, it says the threshold for start of sweating is increased. No, because you want sweating to occur, so the threshold should be decreased, right? So E is incorrect. So C and D statements, if you go through, okay, so they are the correct statements over here. So the right answer should be option 3. Okay, so a little bit if we see in more detail. So human studies have demonstrated that the process of heat acclimatization will begin within a few, that is 3 to 5 days of regular exposure to an exercise in the heat, with most adaptations complete within a 14-day period. So the notable changes are an increase in plasma volume, decrease in heart rate, core temperature during exercise will you have to maintain, so it will decrease, increase in sweating rate and initiation of sweating at a lower body temperature. Okay, as well as there should be redistribution of cardiac output. So there will be increase in blood flow to the skin so that more heat is released. Okay, so that is why if we go through these statements, A and E is incorrect while D is correct. Right? High output for longer periods, sweat glands will sweat. So D is correct. And as I said, it should be vasodilation. So A and E is not correct. There is a significant lower sweat sodium ion concentration for a given sweat rate during heat acclimatization. So this suggests that heat acclimatization increases the sodium ion reabsorption capacity of human endocrine sweat gland. So B is incorrect. So as soon as your body's internal temperature start rising, the hypothalamus will tell the sweat glands distributed all over your body that it's time to start cooling you down by producing sweat. So C is again correct over here. Okay. So if you see this flow chart, you can see how the heat is being, the body acclimatization to the heat. So if there is increase in body temperature, blood vessels will dilate. So there will be heat loss, sweat glands will secrete fluid, evaporation will occur, and then heat will be lost from the body. So the body will return back to the normal temperature. If the body core temperature falls, so the blood vessels will constrict. So that heat is conserved, sweat glands do not secrete fluid. So sweating will decrease and shivering will occur, that is involuntary contraction of muscles that will generate heat. So that will try to increase the body heat. So it will warm the body. So in a way what will happen? Heat will be retained in the body. So body temperature will increase and return back to the normal. Okay. So that is how you solve this question. This is also comparatively a easy question. Next, question number 703110. This is again from excretory system. So the glomerular ultrafiltration coefficient Kf can be changed by the mesangial cells, producing a decrease in Kf, largely due to reduction in the area available for filtration. So if surface area decreases, Kf will decrease, it is saying. The following statements are made about some agents that affect this mesangial cells, that alter the surface area. A. Norepinephrine causes contraction of mesangial cells. B. Angiotensin II causes relaxation of mesangial cells. C. Histamine causes relaxation of mesangial cells. D. ANF causes relaxation of mesangial cells. So again here you should know what are vasodilators, that dilate the blood vessels, and what are constrictors. So which of the following options represent the combination of all correct statements? Again, it's a simple question, just that you should have some information about various vasodilators and constrictors. You can try and tell me. Definitely we all know about angiotensin 2, right? Angiotensin 2, is it a vasodilator or a constrictor? It's a constrictor, right? So it will basically not relax the it will not increase the surface area. Okay, so B will be incorrect. So if you see B is incorrect, means 1 and 2 will not be the options. Now we have to see between C and D and A and D. So what we can do over here is, we need to check between, so D is present in both, means D is a correct statement. So even if you don't know, okay, and you know one, so I'm just helping you out how to solve this type of questions. So we ignored 2 of them. Okay, now let us see. So D, it says it is a correct statement. Now we have to choose between A and C. Okay, so that this is how you can try to solve some questions where you're not very sure of the answers in statement based questions. So between A and C, let us read the statements. Norepinephrine causes a contraction of mesangial cells. Yes. So norepinephrine, basically here you should know that it contracts the mesangial cells. Okay, so A will be correct. Clear? So A and D should be the right answer. The option 4 will be correct, while histamine will not cause relaxation of mesangial cells. Okay. So you can see I have shown you a table. Agents that causes contraction or relaxation of mesangial cells. For contraction you have endothelial cells, angiotensin 2, vasopressin, norepinephrine. Okay, so we see norepinephrine causes contraction. Thromboxane A2, histamine. So all these are they will contract the mesangial cells. Okay, so histamine will also contract, so surface area will decrease. Norepinephrine also contracts, surface area decreases. Angiotensin 2 also contracts, surface area decreases. While if you see ANF or ANP, dopamine, prostaglandin, cyclic AMP, they will all relax. So as a result, the surface area will increase. Okay, so ANF will increase the surface area. So norepinephrine causes a decrease in cellular surface area of glomerulus. That is consistent with mesangial contraction, so A is correct. Angiotensin 2 also induce contraction of mesangial cells. So B is incorrect. Histamine contracts mesangial cells and glomeruli via an H1 receptor. So an effect that is dependent on extracellular calcium entry. So C is again incorrect. ANF dilates arcuate, interlobular and proximal afferent arterioles. So basically it will relax the mesangial cells. So D is again correct. So this table is very important. Okay, make a note of it. So that many times same type of questions are asked. So you can use this table to know which are constrictors and which help in relaxation of the mesangial cells. Okay, as well as you should also know how it controls the blood vessels. So that was 703110. Now we move on to the next question, that is 703111. So this we can say it is related to the circulatory system. Changes in plasma osmolarity and extracellular fluid volume affect thirst by separate pathways as given in the following statements. Hypertonicity leads to osmoreceptor activation, giving rise to hypothalamic control of thirst. B. Hypertonicity leads to baroreceptor activation, giving rise to hypothalamic control of thirst. Hypovolemia leads to activation of baroreceptor and angiotensin 2, giving rise to hypothalamic control of thirst. Hypovolemia leads to osmoreceptor activation, giving rise to hypothalamic control of thirst. Okay, so it is like you have to know about the baroreceptors, what it does, the osmoreceptors. Okay, so it is a mix of these topics. So which of the following options represents combination of all correct statements over here? So the right answer for this question is option 1. Okay, A and C will be correct. Let us see these statements. Statement A is correct as hypertonicity, means there is high plasma osmolarity. So that will activate the osmoreceptors, definitely if the osmotic levels are high. So osmoreceptors will get activated, so it will signal the hypothalamus. So there is less water, right? When hypertonicity means water is less, solutes is more. So there should be sensation of thirst. So it is a correct description of how osmotic changes will affect thirst. Clear? Okay, so hypertonicity means osmotic levels are high. Then, so A is correct. B, it says hypertonicity leads to baroreceptor. Now, baroreceptor is for pressure changes. How will it get activated? Okay, and it will not, so it will not further get activated, the baroreceptors. So B should be incorrect over here. So statement B is incorrect as hypertonicity does not primarily lead to baroreceptor activation. So these baroreceptors are more sensitive to changes in blood pressure and volume rather than osmolarity. Therefore, this statement is incorrect. Statement C talks about hypovolemia, that is reduced extracellular fluid volume. Okay, so what it says, hypovolemia or reduced extracellular fluid volume leads to activation of baroreceptor and angiotensin 2, giving rise to hypothalamic control of thirst. So if extracellular fluid is decreasing, okay, so that time what happens? Baroreceptors will get activated. So you can see if extracellular volume decreases, pressure changes occur. Okay, so as a result, baroreceptors will get activated. So that will lead to the production of angiotensin 2. Here you can see if you follow this flow chart, so angiotensin 2 levels will increase. Okay, so both of this will lead to causing thirst via the hypothalamus. So C is incorrect. So it is a correct description of how volume changes affect thirst. Because if volume decreases, okay, the pressure gets altered. Venous pressure decreases, venous return decreases, less blood. Arterial pressure decreases, cardiac output decreases. Okay, that will in turn lead to increase of angiotensin 2. Now statement D, so C is correct. D says hypovolemia, that is decrease in extracellular fluid, lead to osmoreceptor activation, giving rise to hypothalamic control of thirst. So again, here the receptor is not osmoreceptors that are involved. Okay, so D will be incorrect. For hypovolemia, for hypertonicity it is osmoreceptor. For hypovolemia it is osmoreceptors. Okay, so it does not primarily lead to osmoreceptor activation, the hypovolemia. Osmoreceptors are more involved in sensing changes in osmolarity, not volume. That is why D is incorrect. Okay, I'm sure everybody has understood this. Go through the statements carefully. Okay, you can follow this flow chart as well. And try to remember what is hypertonicity, which receptor gets activated, what is hypovolemia, okay, which receptor gets activated. Now let's move on to the next question. So this will be the last question from evening session. 703112. So certain statements are made below about hemoglobin. HbA1c has glucose attached to the terminal valent in each beta chain. B. NADH methemoglobin reductase system in RBC converts methemoglobin to hemoglobin. C. Oxygen binds to the Fe2+ in the heme moiety of hemoglobin to form oxyhemoglobin. D, the affinity of hemoglobin for oxygen is much higher than that of its affinity for carbon monoxide. So which one of the following options represents combination of all correct statements? Now here, this question is, if you go through the statements, so you will see that statement, okay, B is also correct, but according to the CSIR answer, they have mentioned third is correct, A, C. Okay, so A is correct, B is also correct, as well as C is also correct. Okay, so but because with B I think A is there, so that is why they have not chosen 1, they have chosen 3, that is A and C, which has both the correct statements. Okay, but B is also correct. So I'll quickly go through the statement. Statement B I have written, it is also seems to be correct along with A and C. But accordingly, answer 3 is mentioned. So in a first reversible step, glucose is attached to the N-terminal valine of hemoglobin beta chain. So it forms a labile skiff basis. So for hemoglobin, we should know the structure. It has 2 alpha and 2 beta chains, right? And each of them has heme, and the center of heme is Fe2+, to which the oxygen binds. So in a reversible step, glucose is attached to N-terminal valine of this beta chain. It forms a skiff base that is rearranged to a stable ketoamine bond yielding HbA1c. So that is how HbA1c is formed. So that is why statement A is correct. Next, methemoglobin is an oxidized form of hemoglobin in which iron exists in the Fe3+ state as opposed to Fe2+. So this state of oxidation will decrease the ability of molecule to bind reversibly with oxygen, and therefore disallows it from being able to carry and deliver oxygen to the tissues. Owing to oxidative stress, the methemoglobin is always being produced within the RBCs, but its levels are kept low by enzymatic pathways that work to reduce the molecule. So the pathway that is responsible for reducing most of methemoglobin back to hemoglobin is a NADH dependent methemoglobin reductase system, where the enzyme cytochrome B5 reductase plays a crucial role in a transfer of electrons from NADH to methemoglobin. So that is why the statement B, okay, that also seems to be correct. Right, so we saw A is correct, B is also correct. C, oxygen binds to iron in the heme, forming an octahedral iron complex. So that we know, so it forms oxyhemoglobin. So again, statement C is correct. Okay, so oxygen binds to form oxyhemoglobin. So A, B, C, we saw it is correct. Now the D says the affinity of hemoglobin for oxygen is much higher than that of the affinity for carbon monoxide. So this is obviously not correct. Okay. Hemoglobin binds CO, that is carbon monoxide, about 100 times better compared to oxygen. So statement D is wrong, as is just the opposite of the sign. So if you go through all the statements, okay, C is also correct. A, C and B, all these 3 statements are correct. Okay, but accordingly, the answer that is given is 3, because both the statements A and C is correct over here. Right, so that's all from here. So if we analyze, so there are 6 part C questions over here, right, and 4 part B in the evening session. So you can see at least around 4 questions are very easy, right, I would say. This question is also easy, just that because they have mentioned 3 correct statements, so it becomes little, you get into a dilemma, like how to select if you are not very sure of the answer. Okay, but yeah, at least 4 questions are very easy. Like here, if you know hypertonicity and the receptor that is involved, so this is easy. This one again, thermoregulation, is very easy. This is match the following, that is very, very simple, and this nervous system question is again very simple. Right? So at least 4 are very simple and quick to solve. So before I end this session, I would also like to mention again about the Josh Patch. So for enrolling into Josh Patch and for any information that you need regarding Josh Patch, call on the toll-free number, that is 1800-1200-1818 or 080-5099-7000. Remember, it is for CSIR NET December 2024 as well as June 2025 exam. Thank you. All the best to everyone.